Hello, we want to talk a little bit about transformation by Agrobacterium tumefaciens, which is a good model organism for gene transfer. The general definition for transformation is the introducing of exogenous DNA through the cell membrane in a foreign organism. What is it good for? Because of transformation, the golden rice could be invented, which is an enormous achievement in transformation. A common problem in Asia was the lack of provitamin A in the food. Scientists inserted their gene for this vitamin production in rice and cured this deficiency. Because of that, over 1 million people could be saved from getting blind. Another big achievement was the invention of transgenic plants which are resistant against pests in corn. This problem occurs often in the modern farming and in the result of pests, big yield losses. With the invention of transgenic plants, the yield of the farmers increased. Why do we use Agrobacterium tumefaciens for the transformation? The general features of this bacterium is that it is gram-negative, aerobe and peritrid flagellated. But its most awesome feature is that it has a T plasmid. This plasmid is inducing tumors in decotyle plants. When the plant is hurt, the plant secretes molecules that the bacteria recognizes. If the bacteria attaches irreversible to the plant, it leads this part of the plant to uncontrolled cell division by introducing a part of its own genome called T plasmid in the plant. That's why Agrobacterium tumefaciens is a good model organism for gene transfer and thus transformation. And now we take a closer look on the structure of the T plasmid.
Here we have the T plasmid. The part from the left border to the right border is called the tDNA. This part will be transcripted and introduced in the plant genome. This is also the part which we can cut out. In genetic engineering we can put nearly any gene in this region, which will be after introduced in the plant, like scientists did with the resistance against pests. Normally in this part is auxin and cytokinin. These two hormones are phytohormones and they are the reason for uncontrolled cell division. Next to it is the opin synthesis. Opins are after the transformation produced by the plant for the bacteria, which is the main nutrient for the bacteria. Next to the tDNA there are more parts. They are necessary for the catabolism of the bacteria, like the opin catabolism, and the origin of replication is where the replication starts. Then additionally we have the Vir genes. The Vir genes are important for attachment to the plant cell and for the development of channels where the tDNA can get into the plant cell. And also the tDNA is always transcripted from the left to the right border.
And now we will take a closer look at the process of the transformation. So here we can see the plant cell, our bacteria, containing the T plasmid, the real G protein, and also the real A protein attached to the membrane of the bacteria. Now, the secondary metabolites of the plant cell are activating our real A protein, which phosphorylates the real G protein. This phosphorylation leads also to the activation of the T plasmid by the real G protein. And now, through this expression, we get this real B part. This is like the bridge to the plant cell. Then we get our tDNA containing our genes. Also, we have real D2, which is attached to the 5' end of the tDNA, and a lot of these E2 proteins. Now our tDNA is guided by our real D2 protein to the plant cell. There, the E2 proteins are attached to it to prevent degradation from side of the plant. Through this compound, it's possible for the DNA to enter the parus of the plant. Now we will have a closer look on how the tDNA will get stably integrated into the random part of plant DNA. It doesn't matter where the tDNA will get integrated, because a random promoter can start the expression when it's once integrated. So our tDNA attaches somewhere on the random part of the plant DNA with two endonucleases, via D2 and via E2. Between these two, a single strand will be cut out. Now the single strand of the plant DNA gets cut out and will disappear. So the next step is the integration of the tDNA on the 5' end and the 3' end to the plant DNA. After also the other complementary strand of the plant DNA is cut out by our endonucleases, the plant polymerase will start to replicate the complementary strand of the tDNA. Once it's stably integrated, the plant cell will express the phytohormones and also the opines for the bacteria. And now the conclusion about the transformation of the Agrobacterium tumefaciens. In nature, it leads to the overexpression of phytohormones and opines. The phytohormones are leading to uncontrolled cell division, which causes tumors. The opines are an excellent source of nutrients, containing only for the bacteria carbon and nitrogen. As we have seen, the tDNA can be cut out and be replaced by nearly every other gene of interest. Through this technique, enormous progress in genetic engineering has been achieved. Until nowadays, it is a very useful and important tool for the transformation in plants.